In this video, we're going to talk about animal embryonic development. Before we talk about embryonic development, let's do a quick refresher on what a zygote is and how it is formed. So a zygote is formed when a sperm fertilizes an egg. And so remember that the sperm is the male gamete and it is haploid, so it has one set of chromosomes inside of it. The female gamete is an egg, and it also has, or it's also haploid. It has one set of chromosomes inside of it. When the sperm fertilizes the egg, it forms a zygote. And that zygote is 2N. It is diploid, which means that it now has a set of chromosomes from the sperm and a set of chromosomes from the egg. Now, the zygote does not typically stick around for very long because the zygote is now going to start going through this process known as cleavage. And cleavage just refers to a lot of mitosis. So remember that with mitosis, you have one cell that produces two identical cells. And then each one of those cells can then produce two more identical cells. So you can very quickly go from having one cell to having two, and then four, and then eight. And so every time mitosis happens, each one of those cells is going to create two new identical cells. And so you end up with this exponential growth of the structure. So and during cleavage, so one important thing, and you can kind of see it on this picture down here, you can see that the zygote is a certain size. With cleavage, the overall size of the structure is staying about the same, but the number of cells is increasing. So each cell is actually getting smaller, because remember, a zygote is one cell. This ball right here is showing us 8 cells, but it's the same overall size as the zygote, but each one of these is now a cell. And then this blastula probably consists of, maybe 100 cells or more. And so each one of these is a little cell, but the overall size is staying about the same. So when the zygote goes through the first couple rounds of mitosis, and each one of these arrows is representing a round of mitosis. So when that zygote goes through one round of mitosis, it's going to form two cells that are identical to each other. Those two cells are going to go through mitosis to form 4 cells that are identical to each other. So they have the exact same genetics, they are diploid, they have those paired chromosomes, etc., etc. Those 4 cells are going to go through mitosis to make 8 cells, which makes 16, which make 32, 128, etc., etc. So we keep getting this doubling of the number of cells until we ultimately get an organism. And in the case of humans, we're made up of trillions of cells. So during embryonic development, starting out, these cells are not specialized. So you can look at these cells here in this picture, and you can't tell if the cell you're looking at is going to turn into a skin cell or a liver cell or a blood cell. At this point, they are undifferentiated cells. And so at the 8-cell stage, which is what we're showing right here, these cells are called blastomeres. So each cell is called a blastomere. Those 8 cells keep going through mitosis until they get up to 32 cells. And it ends up forming a solid ball of cells. And it's not shown on this picture here, but it would be kind of between this stage and the blastula stage. And so this morula is just a solid ball of 32 cells. Once again, these cells are not yet differentiated. then mitosis keeps occurring over and over again until we end up with this kind of ball of cells called a blastula, which we can see right here. And in some animals, it is hollow, which we can see here. So in humans, it's hollow, but it's not hollow in all animals. And just to reiterate that we are talking about embryonic development here, okay? So you've probably heard of an embryo. So basically a zygote, when it starts going through mitosis, forming this multicellular ball of cells, really, that ball of cells is called an embryo. And then that embryo, those cells eventually are going to start differentiating and specializing in order to form the specialized cells and tissues that make up an adult organism. At some point during this embryonic development, this ball of cells, this blastula that we see here, is going to turn into a gastrula. So this blastula turns into a gastrula. And basically what happens is that this ball of cells, one side of it is going to start pinching in. And so this is a cross section. So imagine that this is actually a solid sphere of cells, but it has cut away a cross section so we can see what's going on inside. And so you can see this kind of gastrulation occurring here. This pinching in of the cells produces something called a blastopore, which we can see here. And the blastopore is ultimately going to form the digestive cavity. And so it doesn't look like a digestive cavity yet, but it's kind of a pre-digestive cavity. And this is called the archenteron. We are also going to start seeing some differentiation occurring, and this happens in the form of germ layers. And we're going to talk more about these germ layers on the next couple of slides. But as these germ layers start to differentiate, ultimately organogenesis is going to occur. And organogenesis just means the genesis or the making of organs. So these germ layers start to differentiate, and by differentiate, we mean specialize. All right, so they're going to change in their shape and function. To start doing what they need to do to be part of a specific tissue or organ. So for example, your skin cells have to have a different structure than the cells that make up your pancreas because they have different functions. And so different genes have to be turned on and off in order to allow those cells to do what they are designed to do. So let's take a closer look at those germ layers. And remember that these are developing during gastrulation. So before organogenesis. But after the blastula has started to kind of pinch in. On the next slide, I'm going to talk a little bit more about cell differentiation. So if you're a little bit confused right now, that's okay. We're going to get there. But for now, let's just go ahead and talk about the formation of these germ layers. Now, some organisms, some animals, are diploblast, and di means two. And these animals, during embryonic development, they form two germ layers. They have an endoderm, which is also called a mesendoderm, and they have an ectoderm. So you can see that on this drawing here. The endoderm, which is this inside kind of tan layer of the diploblast. Remember, they've got two layers. And then the ectoderm, which is the outer layer. Now they do have what looks like a middle layer, but this is a non-living layer. So there are not living cells in this layer. So it's not considered a true germ layer. And organisms that tend to be diploblast are ones that are going to have that radial symmetry, which we can also say biradial and rotational symmetry, but those things like our sea stars, jellies, echinoderms, etc., etc. Now, triploblasts, so tri means three, and so they are going to have three germ layers. They have that same endoderm and ectoderm that we saw with the diploblasts, but they have this kind of third middle layer called the mesoderm, which you can see right here. And we see triploblastic embryonic development in animals that are bilaterally symmetrical. So these are going to be things like our insects, mammals, reptiles, amphibians, birds, etc., etc. So let's take a minute to step back for a second and take a look at something called cell differentiation, which you could also think of it as cell specialization. So remember that we said that we basically start out as a zygote. Remember, a zygote is just a fertilized egg that it's diploid, it's got those 2 copies of chromosomes, and it's made from fertilization. So we had our sperm, fertilized our egg in order to form a zygote. Now, the zygote is completely undifferentiated, meaning that it is just a cell, but the cell essentially is going to be making all the other cells in the body of this organism. Because remember, the zygote is going to go through mitosis over and over again. And so that one cell is going to form 2 cells. 2 cells forms 4 cells. 4 cells forms 8 cells, et cetera, et cetera. And so this mitosis is going to keep happening over and over again. And that's where we ultimately end up with a morula and then a blastula. Now, each one of these cells, up to a certain point of differentiation, this is what we call an embryonic stem cell. And what this means, we actually, embryonic stem cells are called totipotent. Meaning that this cell is able to ultimately turn into any other cell in the organism's body. So let's pretend like we're talking about humans here. So these totipotent cells are totally undifferentiated. Meaning that the certain genes that are involved in specialization have not been turned on or off yet. So this cell could potentially turn into any other cell in the human body. At one stage, for example, let's go back here and take a look at this 2-cell stage. We could technically separate these 2 cells. And each one of those could then start going through mitosis in order to form its own embryo. And this is what happens with identical twins, where these embryonic cells, at an early stage in embryonic development, they separate from each other. But because the differentiation and specialization has not occurred yet, each one of these cells that branches off or separates off is able to develop into its own organism that's going to be genetically identical to the other cells that it broke off from. Now, at some point, these embryonic stem cells are going to start differentiating. And differentiation just means that gene expression is altered. So different cell types express different genes. All right, because remember, each one of these cells has the exact same genetic material inside its nucleus. So all of the genome, so all of that genetic information is inside each one of these cells. But for example, a skin cell needs to be able to produce a lot of keratin, and keratin is a protein that helps to give your skin cells some kind of structure and toughness to it. But the cells that make up your muscles, they don't need to make keratin. They need to make different proteins, such as actin and myosin, which is needed to allow your muscles to develop and then contract. And so the genes to form actin and myosin are going to be turned on in muscle cells, but not in skin cells. Whereas skin cells are going to have the gene for keratin production turned on, but muscle cells are not going to have the keratin gene turned on. And so this happens when the embryo gets to a certain point. And so our first kind of round of differentiation are going to be those embryonic germ levels. All right, so this is where we're going to get the ectoderm, the mesoderm in the triploblasts, and the endoderm. And so the cells that are making up these germ layers have started on the path to differentiation. So certain genes have started to be turned on and other genes have started to be turned off. But they're still not totally differentiated yet. It's kind of like if you were to let's say you're on a highway, driving down the highway, and you take an exit to go to your house. You're not at your house yet, but you've made a turn or you've started down the pathway to get closer to your house. Now, the cells within these germ lines are then going to continue kind of branching and developing into these different types of tissues and cells. So for example, the ectoderm is going to start developing into things like skin cells or the nervous system. The mesoderm is going to turn into things like the blood and muscles. And the endoderm is going to turn into things like your other visceral internal organs. So things like the pancreas or the lungs. And then even within these kind of differentiation pathways, remember the cells are, those Hox genes really are turning other genes on or off. And so, for example, your skin is made up of multiple types of skin cells. So there are things like keratinocytes and melanocytes, et cetera, et cetera. So I'm not going to go through all of them, but you can see how we have these kind of branching pathways of differentiation. And so with those germ layers, we're kind of taking that first step towards differentiation in order to take these stem cells and ultimately turn them into the specialized cells needed to make an adult body. All right, so now that we have that little aside, so let's come back and take a look at these germ layers again. So the endoderm is going to turn into the lining of the digestive tract. And remember, your digestive tract includes things like your mouth, esophagus, stomach, intestines, etc., etc. Also, the lining of the respiratory tract. So this is going to be things like your trachea and bronchioles into the lungs, etc., etc. The ectoderm turns into the covering of the body. So, for example, with humans, we're taking a look at epithelial tissue, which makes up the epidermis, or the surface of our skin. The central nervous system also develops from this layer, this germ layer as well. And your central nervous system consists of the brain and spinal cord. And then the mesoderm is going to ultimately produce the muscle tissue, other connective tissues, which includes your skeleton, which are your bones and cartilage, and also your blood, and the viscera, which is kind of a generic term for your other internal organs.